Hello, good morning, everyone. This is Elena Minanes at System Biosciences. Today we'll be talking about harnessing the power of extracellular vesicles as biomarkers in disease states. So I will start with a brief introduction, and then we'll go over through two papers where they've assessed different types of biomarkers, protein biomarker for spinal muscle atrophy, and also a microRNA signature for multiple sclerosis disease states. And then we'll dig a bit into how to harness the EVs as biomarkers, best practices, MISEV recommendations from the International Society of Extracellular Vesicles, challenges that one faces, and our latest product to get better and cleaner preps and improve biomarker discovery, that is the ExoQuick Ultra. So, as you may all be familiar with this slide, exosomes are extracellular vesicles of endosomal origin. That's the main difference with other types of extracellular vesicles. They are between 30 and 100 nanometers, whereas microvesicles can go from 100 to 1,000 nanometers. They have different markers, and exosomes are normally generated by inbudding of the cell membrane. They will mature in early endosomes, go into multivesicular bodies, and then the exosomes are released to the extracellular milieu. So here you can see in electron transmission microscopy image how the exosomes are loaded in the multivesicular bodies, and then once the exosomes are loaded into the MBVs, the membrane can dock with the cell. The MBVs can dock with the cell membrane and release the exosomes to the extracellular milieu. Once they're in the extracellular milieu, these exosomes can be uptaken by either neighboring cells or actually distant cells if they go, for example, into the bloodstream. And the mechanisms by which the exosomes can be uptaken are very diverse. There's fusion mechanisms involved. They can also be uptaken by endocytosis, and also ligand receptor interaction. And this one is particularly interesting if we are thinking of, for example, engineering and trying to target exosomes for therapeutic means to a particular cell type. So, as I said in the beginning, the typical exosome size ranges from 30 to 100 nanometers. They're secreted by a wide range of cells, and their composition is going to depend very strongly on the cell type of origin. There's different biomolecules in them, proteins, RNAs, both coding and non-coding RNAs. And the publications in the exosome research have increased steeply in the past years. And this is mainly due to the fact that they have been discovered as very useful biomarkers in different diseases. For example, this is already recent reviews of the role of exosomes and extracellular vesicles as biomarkers in cancer, not only in cancer, but also in neurodegenerative diseases. And we'll go actually into two of the papers that are here, the two scientific reports in a few minutes, and many other diseases. So why EVs are such good biomarkers? So the main reason is that they're present in serum, in plasma, in most biofluids that one can think of: urine, seminal fluid, CSF. And they're not only viable as biomarkers for diagnosis or prognosis, but even also for prediction of response. Not only this, one could also envision a situation where after assessing a biomarker and identifying a given microRNA or protein that needs to be modified, one could isolate exosomes from these given patients, modify them with the appropriate siRNA or protein, and then put them back for treatment. So let's start with this paper where they actually assessed a biomarker for spinal muscle atrophy. This was published in scientific reports recently, and for those of you that are not familiar, spinal muscle atrophy is caused by homozygous mutation of the survival motor neuron 1 gene. It's a disease where the severity inversely correlates actually with the amount of SMN protein that is being produced. It's a very debilitating neuromuscular disorder, and there's been antisense therapy approved recently, but one of the main problems in SMA therapeutics is to stratify the patients and assess the level of expression of the endogenous SM protein. So in this study, the authors showed that the levels of SMN protein that is naturally released in exosomes, and that correlates with the severity of the disease. So here is a scheme of both of the genes that code for SMN protein. There's two genes, SMN1 and SMN2. The mutation in SMN2 leads to lower levels of SMN, induces skipping of exon 7, and leads to reduced levels of the SMN protein in patients with SMA. So the first thing authors did was to assess expression of this protein, the survival motor neuron protein, in different cell types. That's the Western blot on the left-hand side. And then they actually isolated cell culture media and just do a TCA precipitate and assess expression of the SMN protein in the cell culture media. And as you can see, they could detect the protein. So then, of course, they went on and wonder, is this protein actually found within the exosomes or extracellular vesicles? So for this, they isolated EVs from cell culture media, and then after characterizing different exosomal markers, they also did SMN immunogold staining to see that actually SMN is found within the EVs. So of course, then the question is, do the levels of SMN correlate with disease states? So they first work with a mouse model of SMA that is a mutant of SMN protein, and they assess levels both in cell lysate and in exosomes of these proteins. As you can see, there is a huge decrease in the content of SMN in the exosomes of these mouse models. Not only this, but they also checked in human-derived fibroblasts of both healthy controls, carriers, and spinal muscle atrophy patients. And as you can see, the carrier already has lower levels than the control, and then in the disease patients with SMA, there is barely any SMN detected. So it seems that the quantity of SMN in exosomes might be useful as a biomarker for SMA. They also went on and actually quantify exosome production in both the mouse model and in cell lines, and they could see a correlation with increased number of exosomes being produced in disease states, but lesser amount of the SMN protein. So in these plots here on the upper left hand, you can see the nanoparticle tracking analysis of EV count for both a normal control-derived human serum-derived EVs and a patient, SMA patient. And you can see that they could detect an increase in the number of EVs. However, although there is a higher number of EVs being released, the content of SMN is much lower in SMA than in controls. So these results suggest that the analysis of the quantity of this SMN protein isolated from serum-derived exosomes, or the concentration of serum-derived exosomes itself, might be a new method to monitor SMA disease and or response to therapy in human patients, as the problems, the symptoms start when SMN levels are below a certain threshold. So that was one example of a protein biomarker that can be used to assess disease states in spinal muscle atrophy. Now we're going to move to the other main component in exosomes, that are microRNAs. And in this paper that was also released at the end of last year, authors assess exosomal microRNA signatures in multiple sclerosis and try to see if these can reflect the disease status. Multiple sclerosis is a chronic inflammatory demyelinating disease of the central nervous system. And there is currently no single definitive test for MS. Circulating exosomes represent a promising candidate biomarker for a lot of human diseases, and of course also for MS, since exosomes contain, as we mentioned, RNA, DNA proteins, and they can cross the blood-brain barrier. The clinical phenotypes of MS include relapsing remitting MS and progressive forms like secondary progressive and primary progressive, being the RRM the most prevalent and comprising over 70% of the cases. So normally within 10 or 15 years of disease onset, the majority of patients with RRMS will transition to secondary progressive MS. That's a phase of the disease that is defined by the gradual clinical worsening and does not respond to any treatment. So being able to stratify patients into these groups, it's clinically very important. And currently there is no effective test for MS assessment. Diagnosis and disease monitoring relies normally on multiple clinical parameters, including clinical examination, magnetic resonance imaging, CSF assessment. So it's there is a big need for easy liquid biopsy diagnostics or markers that can stratify these patients. So here, authors hypothesize that serum exosomal microRNAs could represent a useful blood-based assay to detect and monitor MS disease. And basically, they start with isolating and characterizing EVs derived from serum, either controls or patients at different states. So they isolated exosomes from 1 milliliter of serum. They actually, before exosome isolation, they treated the serum samples with RNAs to remove any unprotected circulating RNA. And then they analyzed by nanoparticle tracking analysis, as you can see on the left-hand side of the screen, and also by transmission electron microscopy. They assess exosomal markers like CD81, CD63, or an ALIX to identify and characterize the EV isolation. They went on to extract RNA. As you can see here on the left-hand side, the bioanalyzer showed a typical peak of between 25 and 200 nucleotide size, and no ribosomal 28S or 18S peaks. They performed the arrays both with and without the RNase treatment, and with these they were able to identify a set of exosomal microRNAs that are differentially expressed between both disease subtypes. They went on to assess ROC curves for each candidate microRNA and AUC measures for each of them in both of the group, RRMS and SPMS. So with this, they had actually a signature of microRNAs, and they realized that by combining a set of the microRNAs, they could increase the power of stratification of the patients. And while several candidate microRNAs have been previously reported as potential MS biomarkers, the majority that they identified were novel, and this may actually reflect the unique constituent profile of exosomes, an exosomal RNA versus free circulating microRNA, and demonstrates that serum exosomal preparations can actually represent a novel source of biomarkers. So if one is thinking of assessing and looking for biomarkers and using exosomes or extracellular vesicles as biomarkers, what are the things to consider? So we typically like to break down the exosome research workflow in three main blocks. That is isolation, characterization, and then applications, which would be the biomarker discovery or clinical applications such as in vivo delivery, for example. So regarding isolation, there is different methods that one can do, but regardless of the method of choice, whether it's ultracentrifugation, precipitation, size exclusion chromatography, the pre-treatment of the sample of the serum or plasma is very important. The main difference between serum and plasma is that plasma is normally considered a more physiological fluid because there is no coagulation induction. There's all of the blood cells and it's in the state as it was in the patient. However, it requires a very wise choice of anticoagulants. Normally, chelating agents as citrate base are the most recommended, as heparin may cause interferences, especially with PCR and NGS downstream. It's also important the choice of pre-clearing. How are you going to get rid of the blood cells, centrifugation, thrombin treatment that will turn the plasma into a more serum-like fluid. And with serum, again, you still need to do a pre-clearing and centrifugation to remove all the debris, and then proceed to isolation. What is the choice going to be based on? I would say most of the cases is based on availability and what the clinical samples that one can get access, but it's also good to think, if you have the possibility to choose, to think of the experimental design. If you are going to be able to treat all the samples equally, plasma requires to be processed within 30 minutes, hopefully between 30 minutes and an hour after drawing the blood, whereas serum can be stored easily at -80 after once it's processed. So the International Society for Extracellular Vesicles in 2014 came up with the minimal experimental requirements for definition of extracellular vesicles, and in the review of this position paper is coming up soon. But for now, what is recommended is that one, to characterize the single vesicles, one should use at least two different and complementary techniques. That is, for example, electron or atomic force microscopy to have an image of the EVs and see the size and the shape. And then also single particle tracking, like nanoparticle tracking analysis, where one can assess both the size and the count. Then general characterization in terms of markers, so at least 3 positive protein markers for EVs, being at least one of them a transmembrane or lipid-bound protein, and or a cytosolic protein, and a negative protein marker, such as, for example, here we can see, such as argonaute or histones, or proteins that are known not to be, or that should not be contained within exosomes. So challenges in the use of EVs as biomarker discovery. Well, if we think of the two main components, RNA and protein, let's start with RNA. So exosomal RNA, as you saw before in the paper we were going through, and as you can see here in this electropherogram, it's mainly comprised of species between 20 and 250 nucleotides. There is microRNAs, there is long-encoding RNAs, mRNAs. One of the main challenges here is, one, the concentration is going to be very low. Two, one needs to be sure that it's not dragging any free circulating RNAs or ribonucleoproteins that may contain RNAs, but that are not contained within the exosomes. Also, it's important to take into account that the makeup of the exosomal RNAs is going to vary a lot between individuals and between sample types. So in this paper, they assess exosomal RNA by RNA-seq from 4 individuals, both from serum samples and urine samples. So you can see that there is a clear distinction between the serum samples and the urine. For example, the serum are much richer in microRNAs than the urine samples, but there is a lot of variability intra-individual. So the number of samples that one uses to try to assess and to come up with a novel biomarker, it's very important. The way one treats the sample, the processing, the isolation, all of this is going to affect the final makeup composition of the exosomal RNA and may actually be affecting the biomarker discovery process per se. This is data that we actually have generated also in-house with our ExoNGS service, where you can again see that the composition of the exosomal RNA varies enormously between different types of samples. If we now move on to proteins, there is a wide range of proteins within exosomes, cytoskeleton proteins, heat shock, enzymes, tetraspanines, signal transduction proteins. So the approaches to assess protein, the current approaches to assess and characterize proteins in individual or actually in any sample, go from the classic Western blotting that is, yes, easy to do, and there's many antibodies available, so if one knows which protein it's targeting, like, for example, in the paper that we just went through, where they just wanted to assess SMN, because SMN is the levels of this protein correlate with the disease, so let's just assess the levels of this protein in different disease states. That's a good enough approach. If we're thinking of biomarker discovery, then Western blotting is going to be quite limiting because it's low throughput. You can only assess ID of known markers, right? Protein arrays has the advantage that, yeah, at least it's scalable and low to mid throughput. And there are many commercially available panels. However, it's expensive. And again, there is a certain bias in the sense that you're only IDing known markers, and there is also a quantitation bias. The most comprehensive approach, that is mass spectrometry, is an unbiased protein quantification, but it is a complex setup. It requires expertise. The cost is much higher per sample, and it's extremely sensitive to contaminating proteins in the sample. And this is just to show how what the typical profile, protein profile in serum and plasma looks like. And as you can see, albumin, it's over 50% of the total proteins. So if you're thinking of assessing a biomarker, this will most likely fall into this pool of containing low abundant proteins that is probably somewhere between 5 and 10% of the total proteins. So it's really difficult to find these biomarkers within these samples with all of this background and contaminating proteins. This is data that we generated actually in-house, where we compare mass spec of either a standard prep or a surface protein capture kit that we have to do a mass spectrometry. And you can see that the percentage of reads that actually map to either apolipoproteins, fibronectin, IgGs, serum albumin, when there is no post-isolation cleanup, it's extremely high. It's over, if you put them all together, it's probably over 45% of the reads. So if you're doing mass spec, that is really expensive, but almost half of your reads are going to all of these background proteins, you may be losing and missing out on important biomarkers. So from this need of a post-isolation cleanup, we came up with ExoQuick Ultra. And ExoQuick Ultra is one of our, it's an improvement compared to our standard ExoQuick EV isolation, precipitation based. You can see here that the EVs isolated with ExoQuick Ultra display a typical EV morphology. These are transmission electron micrographs of EVs isolated from human serum. And the one on the right is just a close-up of the one on the left. With ExoQuick Ultra, we have actually managed to reduce significantly the amount of albumins and IgGs, as you can see in this Western blot for exosomal markers like CD9, CD81, and HSP70. The detection of this exosomal marker is highly increased compared to other commercially available kits, and also compared to ultracentrifugation, because since we are getting rid of albumin and IgGs, when you're now loading 5 micrograms of protein in your Western blot, you're actually loading many more EVs than with other EV preps. Not only this, but we actually have managed to increase the yield, and this enables you to isolate more EVs per both normalized input volume and per milligram of protein, compared to either other commercially available kits or compared to ultracentrifugation. So how does this work? It's a very easy protocol. It's based, again, on our ExoQuick precipitation isolation. The only difference is that once you have precipitated your EVs, you will do a cleanup that is column-based, and is just an extra 10 minutes on your isolation procedure. So the advantages of ExoQuick Ultra is that really it yields cleaner preps. It increases the yield of EVs that you isolate, starting with the same amount of material compared to either ultracentrifugation or other kits. This actually enables a better biomarker detection. It's actually very fast, and it's also very cost effective. So I will now open up for questions. I have here the scientist that actually developed this kit, Beekeeper9. And if you can just type your questions into the chat box, we'll try to go through them. And if we don't get a chance to reply to all of them, we'll just try to get back to you by email. So one of the first questions: do EV secrete nucleic acids to the bloodstream? I'm actually not sure if EVs have the ability to secrete. I don't know, maybe.
So far, which is known that cells are secreting nucleic acids to the bloodstream. They are known as exRNA. EVs has their nucleic acids inside the EVs. They shouldn't secrete them, or at least it's not something that is known in literature.
What size of vesicles are you recovering with the ExoQuick Ultra?
The size is approximately between 70, 80 nanometers up to, I would say, 200 nanometers. Depends on the prep.
Okay. Another question: what is the best way to isolate EVs from culture media? So we do have a version of ExoQuick Ultra that is specially designed for tissue culture media and fluids that are less abundant, like CSF or urine. That is the ExoQuick Ultra TC. And yeah, it's also always good to think what are the downstream applications that you're gonna use with your exosomes when you're thinking of the method of choice. some people are asking if the slides would be available. If you want to have a copy of the slides, please send us an email to tech@systembio.com, and I can get you a copy. Can ExoQuick be used for functional EV isolation?
Yes, definitely. This is the purpose of ExoQuick Ultra, to study functional for functional studies.
What would be the best system to use when trying to obtain enough small RNAs from EV for library preps for NGS? So here, I mean, I think especially for small RNAs, it's important, and there is quite a few, I mean, in this paper they do it, but there's quite a few papers where they do the RNase treatment. I think it's quite important to have this control and to make sure that the RNAs that you're detecting are inside the exosomes. But I would say, I mean, ExoQuick Ultra would be a very good choice in the sense that you're getting rid of background proteins, so you're having a much cleaner prep, and you're also increasing the yield. So how many milliliters are required for human serum?
I think it is, you can start with 100 microliter for ExoQuick precipitation and follow with the ultra cleanup. An average is what we are using here is 250 microliters, and it works very well, and the yield is very high.
So another question here is whether ExoQuick has high throughput format like 96 well format for serum. We don't have this yet. I mean, we understand that for clinical applications, it makes things much easier. But actually, it is much easier to tackle many samples if you're doing a precipitation-based approach than if you have to do ultracentrifugation or other approaches. I don't know. What is the range or heterogeneity in EV size in different samples? I think, I mean, it's, yeah.
EV's population is very heterogenic. They are including exosomes, microvesicles, which all vary in size from 70 to 150, 250 nanometer. There is no one format for example, to say these are exosomes and their size is in this range, or these are microvesicles and they are in this size. They are all extremely heterogenic population.
Do you treat the exosome prep with protease before isolation to eliminate contaminated proteins? No, I mean, well.
No, we do not use proteases, not before isolation, not during the isolation. In general, I don't think that adding proteases will eliminate contaminating proteins, because proteases are proteins, and I don't see any good idea why to add more proteins into your isolation procedure. In general, we're using ExoQuick. It is polymer-based isolation system, and it protects actually your proteins during the isolation from the degradation or anything else.
How would you dissolve, another question, how would you dissolve an exosome pellet in PBAs as they aggregate generally? Sometimes pellets can be difficult to dissolve.
It is sometimes hard to dissolve the pellets from the ExoQuick precipitation. It takes time. Just spend 1 to 2 minutes on pipetting. It will be fine. You will be able to dissolve the pellet. If they are aggregate, From what we noticed, if you dissolve your pellet in PBS, they not immediately aggregate. If you will leave them on at 4 degrees for weeks, they will start aggregating. That's correct, but you still have time to work with them in PBS.
Okay, we have one next question. Do you recommend to confirm EV staining markers with TM? I'm guessing you're thinking of immunogold staining with TM. I mean, I think that's a very particular application when you may want to do it with a particular biomarker once you have identified. But I would say, I mean, for general exosomal or EV markers, Western blotting is just fine. Yeah, once you identify a given biomarker, if you really want to show that this protein is inside exosomes, then yes, you can use immunogold staining to assess this. How stable are the exosomes?
The question is, what is the time until they start aggregating, or the question is the stability in the solution in terms of pH or in terms of different proteases. So the question for a first aggregation part, if you freeze your sample and you don't have, and you aliquoted it before, there is no repeatable freezing and thawing cycles, so they are pretty stable in terms of aggregation. I wouldn't recommend to store them at 4 degrees for a long time. In terms of stability for pH or for proteases, they are very stable molecules.
Can ExoQuick Ultra be used for isolating specific size of EVs?
ExoQuick cannot discriminate between the different types of EVs. It isolates heterogenic population of EVs. And as well as ExoQuick Ultra, which is the more clean isolation way of EVs, again isolates heterogenic population of EVs.
Okay, I think we will close now. There might be a couple of questions left. We'll go through them, and if we have not replied to your question, we'll just send you an email and let us know if you have any further questions. Thanks for joining. Bye.